So last time we were talking about the different models that have been used to explain motivation. We were talking about homeostatic models, which suggested that there was a system that tried to keep a set point, a balance point in your various motivational systems. And then we also talked about a newer model, which is an incentive motivation model, which says that you have a want and a like system. And that want and like system are connected to dopamine and endorphin systems in your brain. So now let's apply some of this to talking about where does that research come from. So Olds and Milner in 1954 were actually conducting an experiment where they were interested in seeing if electrical stimulation inserted into the hippocampus could stimulate memory in rats. So would they learn faster if they were able to, every time they pressed a lever, as you can see here in the diagram, could they stimulate their learning? But they actually, unfortunately, they missed the hippocampus, and instead they put it into this structure called the medial forebrain bundle, what we sometimes call the reward pathway. That medial forebrain bundle, whenever the rats would press it, they would get this intense psychological stimulation where they would want to keep doing that same activity. So these rats would sit there and press that lever for 10 or 20 or even 40,000 times. They would not eat, they would not drink. All they wanted to do was press this lever. It was the first evidence that suggested that there was a specific pathway related to addiction. So we now know a lot more about the pathway, and we know what's going on, as we kind of talked about with incentive motivation. So here's a ghosted in image of the human brain and a couple of structures here. We're going to start in the front. So this is your prefrontal cortex, and your prefrontal cortex actually sends a signal all the way back to an area called your ventral tegmental area, your VTA. And your prefrontal cortex is basically almost like a brake. So it gives you impulse control and it turns off, for example, stimulation from the ventral tegmental area, which gives you that want sensation. So every time you think, oh, I really want to eat that entire carton of ice cream, your prefrontal cortex, right, sends a signal back to your ventral tegmental area saying, don't eat the entire carton of ice cream. Most of the time, right? Okay, but then right in front of the ventral tegmental area is your hypothalamus, and we've talked multiple times about sort of the various roles that the hypothalamus plays in motivation, and we'll talk more about it as it relates to appetite in just a second and eating. Then you've got just behind your ventral tegmental area, which is here in orange, is your substantia nigra. So remember, your ventral tegmental area is a dopamine containing area, and your substantia nigra is also a dopamine area. But these two dopamine pathways do different things. The ventral tegmental area is the dopamine pathway for motivation, and the substantia nigra is the dopamine pathway for movement. And it makes sense that anything you're motivated to want, you're going to need to move towards, right? If you want food, you've got to go look for it in the kitchen or at McDonald's or wherever you're going to go. So of course you would want those two areas to be really close to one another, and they probably grew out of one another. Then up in the front, you've got your nucleus accumbens. So this is the teal colored structure. The nucleus accumbens is that like sensation. So it releases and responds to endorphins. And so normally, as we talked about, the ventral tegmental area stimulating want and the nucleus accumbens stimulating like are coordinated together, but we talked about a few examples where those maybe don't match up. And then you've got the amygdala in green, which is for fear sensations, and then you've got the insula, which is for aversions. So let me give you a good example. Foods that you don't like, that you dislike because of flavor, texture, odor, that's signaled by the insula. Whereas if you're afraid of snakes, that's signaled by the amygdala. Okay, so these basic structures are what's critical in a lot of motivational processes, but let's apply it to hunger and eating in particular. Okay, so your GI system, first of all, the glucose that's contained within your cells is really what determines your level of hunger. So cells that don't have enough glucose in them will stimulate through various processes for you to be hungry, and cells that have too much glucose in them will signal that you're full. Okay, but your stretching of the stomach, the distension of the stomach actually does trigger a sensation of satiety. But it's not because of the physical stretching that stretching of the stomach actually causes you to release a hormone called CCK. So as those cells in the stomach stretch, they release cholecystokinin or CCK. That then triggers the valves in the stomach to close, so your stomach starts filling more quickly, and that gives you that further distension, which gives you more CCK release. So you get this positive feedback loop where then it starts, makes you not hungry anymore. But we do know that people who eat more slowly do show decreases in meal size, but I think I mentioned this, we've got to be careful about some of that data because some of it came from that lab at Cornell, which a lot of his research has been brought into question. But there are lots of things people can do. We can do gastric bypass surgery. Gastric bypass surgery is effective at decreasing mortality as long as it's done before you're the age of 40. People that have gastric bypass in these large studies after the age of 40, it seems like the damage has already been done. Their rate of heart disease, their rate of heart attacks, their rate of cancer doesn't go down, actually. So they're just like a placebo group that didn't have the surgery at all, or a control group. So, and of course, right, there are lots of problems with gastric bypass. It's got a high rate of infection. About 5% of people will actually have complications related to infections or malnutrition. And of course, right, some people, you can go back to your old eating habits. So it requires that you work with a nutritionist and a psychologist to make sure that you're maintaining your smaller, more regulated eating habits, and that you're not getting malnourished because you can only eat small meals. But you also have a stomach hormone called Ghrelin or Ghrelin. So Ghrelin is related to growth hormone, and Ghrelin actually stimulates short-term feeding. So it sort of makes you have this intense temporary, like, okay, I need to eat a little bit more, and then it dissipates. So you've got these two hormones in your stomach that are regulating whether you're eating or whether you're full. Then you've also got a long-term satiety mechanism. So CCK is what makes you have that temporary, okay, I'm full now for an hour and a half. Your stomach will eventually fill, it'll then empty, you would be hungry again. Leptin, on the other hand, released from body fat tissue, from adipose tissue, triggers long-term satiety. So people with higher levels of body fat, animals, for example, with higher levels of body fat, that releases leptin. That leptin goes up into the hypothalamus, and that signals them to stop eating. So they have this sort of homeostatic-like mechanism where their leptin levels regulate how much they're eating. But clearly that isn't happening in everybody, and clearly there are mechanisms for people who are morbidly obese, for example, where either maybe they're insensitive to leptin, maybe they have a receptor difference, so maybe their leptin receptors are not very sensitive to the leptin they're releasing. A couple of different studies have shown various methods by which people may not respond to those normal hormonal signals. We also know that if you look at rats, that in fact the hypothalamus is critical in their feeding intake. So these are two sets of rats. One had a sham surgery, which means they're put under anesthesia. We make a small surgical incision into their skull. We put an electrode down into their hypothalamus, but we never turn it on. So they've had everything else done to them. They're like a control group. They have had everything else done except for the lesion. But another group of animals had a lateral hypothalamic lesion, and what that found is that the lateral hypothalamus seems to be critical for your normal eating behavior. So without it, when it's been destroyed, you stop eating. And in fact, actually, these animals will lose a significant amount of weight, as you can see here around day 5 after surgery through day 10. And in fact, if they're not force-fed, they will starve. So these animals have to be fed in those first 5 days, but after that, they seem to recover. But you can see their body weight has dropped from about 390 grams at the very start, right? And then it's here, and the initial part of the surgery, they've gone to about 360. So they're losing about 10% of their body weight through just a simple lateral hypothalamic lesion. And what's interesting is that you can actually pick apart the different pieces of their eating behavior. So what is it about food that they're not liking? And it seems to be that they don't like the texture, the flavor, the taste. They don't get any enjoyment out of it. They basically eat because they have to. Essentially, you've retrained them in these 5 days to eat again. And now they just eat it, but they don't seem to enjoy it. Okay. So the lateral hypothalamus is important for the stimulating your food intake. And without a lateral hypothalamus, then you start to lose weight. Okay. The reason for that is, so here's all those various nuclei within the hypothalamus. So, and we've only talked about a few of these, and I'm going to sort of hit on a few as we talk about them, but all of this are structures within the hypothalamus, okay? And of course, the thalamus is right here above it. So you can see here we have a coronal cut. So we've sliced the brain across the front. So we're zooming in on this purple box right here. So the lateral hypothalamus, again, of course, is on the sides. It's the green area right here. And the lateral hypothalamus releases 4 hormones that seem to be critically involved in maintaining your normal feeding behavior. Neuropeptide Y, or NPY, agouti-related peptide, or AGRP, orexin, and melanin concentrating hormone, or MCH. Orexin, interestingly enough, has also been implicated in narcolepsy, as we'll talk about when we get to our chapter on sleep. So some of these hormones may be doing multiple things across different brain systems. Okay, so we were just talking about the lateral hypothalamus, and the lateral hypothalamus is important in stimulating hunger, but the ventromedial hypothalamus, or VMN, the ventromedial hypothalamus is important in turning off hunger. It's your satiety mechanism, okay? So again, ventromedial, medial means towards the middle, so this is towards the middle of the brain, and ventrum means towards the stomach, so that would be underneath in a human, that would be underneath the chin of a human. That's the ventral surface of your hypothalamus, okay? So when we do a ventromedial hypothalamic lesion, a VMN lesion, okay, what happens is these animals keep eating. So my MH lesioned or VMN lesioned animals here are in light purple, and you can see that they keep gaining weight. They started around 200 grams, and they go up to about 350 grams in about 5 weeks. So they're almost doubling their body weight in 5 weeks' time. Whereas the sham surgery animals, remember, they've had the anesthesia, they've had the surgical incision, they've had the electrode put in but never turned on. So we're not damaging other neurons. So any neurons that were damaged because of the insertion of the electrode were happening in both surgeries, okay? So these animals with the ventromedial hypothalamic lesions are almost doubling their body weight. So clearly the ventromedial hypothalamus is important in regulating how much you eat. And here's an image of these 2 rats. You can actually see where I know it's kind of pixelated, but you can see where these rats had the incision right here in the top of their head. This is a sham surgery animal, so this is kind of a traditional Norway rat. This is one that has had the hypothalamic lesions, and they're gained significant amount of weight in those 5 weeks. So, like with the lateral hypothalamus, the ventromedial hypothalamus has a few chemicals that are involved in satiety. One is the alpha melanocyte stimulating hormone, or alpha MSH. This is also the hormone that helps to regulate melanin, which is your skin coloration. You also have another one I briefly mentioned last time called the cocaine and amphetamine related transcript, or CART protein. Then also CCK, that cholecystokinin we talked about earlier, leptin, and insulin. All of those go to this ventromedial hypothalamus right here and tend to turn off hunger, either in the short term, for an hour or so, or in the long term, over days or months. So that's a lot, right? We talked about 9 different hormones that are regulating hunger and satiety. And in fact, there are over 50 hormones that we believe to be involved in hunger and eating. So this is my attempt to try to make sense about, well, what are some of the complexities here that's happening? So everything in red is a satiety mechanism, okay? So all these red arrows are things that are turning off hunger, okay? So leptin and insulin, as we mentioned earlier, if your insulin levels goes too high, it makes you not want to eat anymore, because it's basically telling you that you're using all of your insulin to move glucose from the periphery to get inside your cells, okay? And leptin is that long-term satiety mechanism. So those go to the arcuate nucleus specifically, okay? So that's another region we haven't talked about, but the arcuate nucleus controls both hunger and satiety in separate areas. So Ghrelin or ghrelin from the stomach and leptin and insulin from your peripheral nervous system and your stomach are coming into the arcuate nucleus in different parts of it, either to regulate hunger or satiety. Okay, so ghrelin stimulates hunger, which then stimulates the release of 2 of those proteins we were just talking about, neuropeptide Y and agouti-related peptide. So that then sends a signal to this lateral hypothalamus, remember, on the side, which is what stimulates hunger, which then triggers you to release MCH and orexins, which trigger you to then start eating, okay? Similarly, those also trigger the release of TSH, which is thyroid stimulating hormone, and ACTH, which is one of your stress hormones we'll talk about in a few weeks, also stimulating hunger. And it makes sense that after a stressful event where you've been releasing stress hormones, you're going to need to replenish your glucose stores because you've used them up. So stress hormones should probably trigger you to start eating. Then you've got within the arcuate nucleus, right, leptin and insulin that are turning off your hunger mechanisms. They're triggering satiety by then triggering the release of alpha MSH and that cocaine and amphetamine related transcript. That goes both to the lateral hypothalamus and also to the paraventricular nucleus, or the PVN, triggering satiety mechanisms, okay? And then lastly, this other one from the stomach, the CCK, is going directly to the paraventricular nucleus. And it makes sense that you may be thinking, oh my gosh, there are just 9 of these, and that's already incredibly complicated. Why do you have so many hormones regulating hunger and satiety? But of course, you want to be careful as an organism that you are making sure that you're eating enough when food is available, and when food isn't, or when there's too much food available, that you can regulate, right? You can satiate yourself and not overeat, especially for a bird, right? Birds who overeat can't fly successfully. So there are lots of organisms that are probably trying to balance their needs, which means if you have some genetic condition that makes you insensitive or not able to release one of these hormones, if you only had one hormone controlling the system would fall apart. So it makes sense that over evolutionary time, we've probably evolved many redundant mechanisms to help make sure you're feeding yourself. If you have any questions, give me an email. Bye.